space crop production facility. It consists of a number of different labs. Across the hall here, we have Greenworks, where a lot of testing, hardware testing takes place. We can get engineers in on site that construct and test any number of things. We can do lighting, plant testing over there. The lights are off right now, that's why it's kind of dark. In this lab, we have a number of walk-in chambers and reach-in chambers. They're environmental grow chambers. They have the ability to adjust the temperature, humidity, and CO2 conditions. A lot of times what we do is we mimic ISS conditions in order to cultivate these plants. These plants are grown like 3,000 ppm CO2, and we want to evaluate how well they do in those environments, and they don't necessarily always like it. So we use these chambers to simulate that environment. After we grow all these plants, are they safe to eat? That's where Mary comes in.
That's where we come in. So we have a team of microbiologists that, support all the horticultural plants and crops that we grow. And our job is to understand the microbiology of the plant, good and bad. So as we know, we all have our own good microbiomes on humans. Plants have the same thing. So these are root-associated bacteria that help the plant. But we also screen everything we grow that is intended to be consumed by the astronauts for food safety. So just as your food that is provided by a company, they do the same thing. They screen it to make sure the consumer doesn't get sick. So we have a crop planned of tomatoes that is going to fly in, I think it's July of this year. So we'll do all that analysis first, and then the JSC medical team will look at our data and say, yes, it's okay to eat. So we have grown I think it's 14 different types of leafy greens and peppers, and they've all been able to consume, the astronauts consume those on the space station. So that's basically what I do in support of the platform.
And all this is to create food stuff for deep space travel, right? So
right.
That the astronauts won't have to take everything, they can bring it along with them and make it happen on route.
Right. So the horticulture side of things is to test a lot of different crops, and from those you select based on how well they grow. So our aspect is, well, let's look at the system and the microbes, and is it also appropriate to fly in a small system like veggie or some other type of system, some of the things that are planned for the future as well. So, and we also are starting to work on the introduction of some good microbes, like a biofertilizer. So that's an area of investigations.
Not just food in the tube anymore.
What's that?
Not just food in the tube. Yeah,
that's right. I mean, they really, I don't know if anybody has seen any of those images of the chili peppers that LaShelle was instrumental in that. Sure. So they really, I think for the most part, really enjoy having fresh, good, safe produce. So.
And talk about in a nutshell how we got the Espanola chili pepper to the advanced plant habitat. It started right here in this lab, in this chamber over here. We screened dozens and dozens of chili peppers to make sure that they were compatible with the environment to which we wanted to grow. Did they have good germination? Did they have the right size? Are they compatible with LED lighting? And we did them in these chambers. So about 3 years ago, we grew chili peppers to the extent, but right now we're testing strawberries. So maybe in 3 years we'll have strawberries, right? So not only did we screen multiple different cultivars, meaning varieties of chili peppers, we used these chambers to make sure that they were the right size. Then we moved over to Greenworks and tested how well Espanola chili peppers grew in an APH analog system. We did that over there. And then from there, it went over to the APH Engineering Demonstration Unit, which is the EDU. And we did 2 tests in there to evaluate its compatibility with the hardware. So before it went to ISS, all that work started here with the people in this lab. That project was also successful because of the seed sanitization methods that were developed by Mary's team here. If they were not not sanitized correctly, we could introduce pathogens like Fusarium into the system that we don't have the ability to clean out. If they're not sanitized correctly, they won't germinate, they won't be viable. So she's here because she's an integral part of our team, just like the things that you see here. So hopefully one day we'll have strawberries there. The next step for these strawberries will hopefully be Chipia. We're currently testing them in the aero gardens that will be used in Chipia, which is a 1-year analog, which will have a food production aspect. A small portion of it, the analog crew will be growing these fruit, hopefully. We're still testing it. But right now we do have a list of leafy greens, tomatoes, and peppers, and leafy greens, tomatoes, and peppers that will also be tested and grown in Chipia. But those crops started right here, right? So, we're
Space
excited.
taco?
So you said,
Oh yes, the
you
space
said they'll be
taco.
in there for 3 years?
Excuse me?
You said they'll be in there for 3 years?
So, the Chipia analog will run for 1 year, and I think in 3-year tests. They'll do it 3 times. The food production aspect of Chipia, there's, I think it's 7 weeks. Don't quote me on this. But it's only a portion of it. Like, they're only going to be growing their crops for certain portions of the 1 year scenarios. But that's the next step for these, hopefully. We worked with the USDA. They gave us a genetically modified plum. It was modified so that it flowered and fruited within a year. And so this work was done over in the SLSL lab. So we tested a number of their different modified lines to see how compatible it was with controlled environments and different CO2 levels. So we have tested trees. Yeah.
So one of the, I'm just looking at the chart.
Yep.
One of the great environmental challenges of growing in an environment like this is water use.
Yes.
Because I see that noted on here. You can't just go turn the hose on them like you can the plants in your garden. So what are some of the engineering processes you go through to sort of decide what plants can survive that?
Well, for this test, we did things like stomatal conductance measurements to evaluate how much water the plant gave off. And what we found was that the genetically modified versions were unable to open and close their stomata well. So they actually utilized way more water than the unmodified versions. So while this is a great construct terrestrially, maybe it's not quite ready for extraterrestrial use because the water use was exponential.
Yeah.
So we just couldn't keep up with it, at the time.
And you have to
6 years
try to
ago.
go through the experiments and go through the process to figure out
To
which
figure
plants are going to be able to make it.
That's one of the things we do figure out in these chambers, which crops are more water hungry than others. So that's one thing we do figure out. Let's see if I got some microgreens to show you guys.
Okay.
So another project that we have, we're evaluating microgreens. Microgreens are small little plants that grow within 7 to 14 days. They're jam-packed with nutrients. They'll be an easy crop to grow for potential crew members to consume. They're of interest because, one, they're easy to grow. They germinate quickly. They have way more nutrients in them per gram than maybe a standard size crop. And if you're short on time, you can sprinkle little microgreens on them. I have a picture of them, so you can't really see them in there. But we have screened over 70 different types of microgreens in this lab, from basic ones like mustard, collard greens, kale, to more exotic things like quinoa, cantaloupe, and even herbs like basil and parsley we've screened as microgreens. And this would be really great to grow it in 7, 14 days, sprinkle it in a taco, sprinkle it in your salad. It's not, our goal is not to make a giant salad. Where our goal is to supplement the diet, to add benefit and psychological benefit to these meals and spice them up and add different textures and flavors.
Yeah, you want nutrients packed into a small space, basically. You want something that's easily transferable, but with a lot of nutritional value, because otherwise it's not going to have the same need to fill the astronaut's needs.
And additionally, we found that the the food that they will leave with, actually the nutrients degrade over time. In about 3 years, a lot of really valuable nutrients like vitamin C and vitamin B1 degrade over time. So we're trying to supplement that vitamins with that. One last thing. So these chambers are great when you have a huge experiment, right? But what if you want to test different light treatments? What if you want to test different CO2 treatments? And we don't have enough chambers for them, right? We have the ability to test 6 different light treatments, 6 different CO2 treatments, 6 different anything treatments with the use of these chambers called the Exciter chambers. They were custom designed and custom built by Lawrence Koss on our team, and they have the ability to have their own micro environmental system in each of these chambers. So a number of researchers have used them to introduce a number of different treatments to plants. And this kind of condenses our science so we can get a lot more science in a smaller footprint.